and send your question via the chat box or use the raise hand features during Q&A session. Ladies and gentlemen, good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Distinguished guests, respected speakers, and also kawan BRIN who are joining us on site and online via Zoom meeting and BRIN Indonesia YouTube channel. First of all, let's thank God for blessing us and allowing us to gather here today in a good conditions. Welcome to the 12th Real Variant Focus Group Discussion, Accelerating Collaborations of Food Research through Nutrifoodsync. Thanks to you all for attending this event, and I'm Mulyani. I'm honored to serve as the Master of Ceremony for today's event. In this occasion, we are truly honored to welcome the Honorable Chairman of the Research Organization for Agriculture and Food, National Research and Innovation Agency, Mrs. Puji Lestari SP, MSc PhD, Distinguished Head of the Research Center for Horticulture, National Research and Innovation Agency, Mrs. Dr. Randa Dwinita Wikan Utami, that also will be served as a moderator for today's event, and also all heads of the research center within the Research Organization for Agriculture and Food, BRIN. Distinguished speakers, Associate Professor Sasya Pramaputri from Osaka University, Japan, Associate Professor Dr. Mahmud Iwan Solihin from UCSI University Malaysia, Dr. Yudhistira Nugraha SPMP, Head of Research Center for Food Crops, BRIN, Dr. Satrio Kridowahono, Head of Research Center for Food Technology and Processing, BRIN, and also distinguished participants. We are delighted to have you here, whether in person or virtually, coming together to share knowledge and foster collaboration for sustainable future. Ladies and gentlemen, for your information, let us share a brief introduction about today's event. RUA Variant stands for the Research Organization for Agriculture and Food Sharing Forum. It is a regular discussion series organized by the National Research and Innovation Agency through Research Organization for Agriculture and Food to strengthen research collaborations, share knowledge, and address strategic issues in agriculture and food. And today marks the 12th edition of RUA Variant with the theme Synergy of Superfoods, Alternative Food and Precision Agriculture for Sustainable Food Security. Ladies and gentlemen, to officially open today's session, we are honored to have Chairman of Research Organization for Agriculture and Food, BRIN. Due to another official engagement, Mrs. Puji will send her messages from recorded.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, good afternoon, everyone. Distinguished speakers, Professor Sastia Pramaputri from Osaka University, Professor Mahmud Iwan Solihin from UCSI University Malaysia, Dr. Yudhistira Nugraha, the Head of the Center for Food Crops, Assalamualaikum warahmatullahi wabarakatuh. On the hard artists and distinguished speakers, professors, distinguished guests and friends across the globe, attending this Professor Mahmud Iwan Solihin from UCSI Head of the Science, adalah moderator the Head of the Center for Agriculture, esteemed speakers, ladies and gentlemen. On behalf of the Organization for Agriculture and Food BRIN, is my great honor to distinguish this across the globe on accelerating innovation of food research through Nutrifoodsync. We are very delighted to be here together with you in this international gathering where my discovery inspired
Something disturbing.
Agriculture and food security and supply this driven by climate change, accelerating innovation of food research through Nutrifoodsync response to mass reports. We are very delighted to be here ladies and gentlemen, inspired by this foreign synergy of superfoods,
Something disturbing.
To the prevalence is contributed to higher rates of non-communicable disease, and suppressed in this industry, especially bioactive compounds, here are more indigenous food sources such as sugars, cereal, legumes, tropical fruit, insects, and fish, offer high nutritional values and for potential for superfood development. Advanced technology such as metabolism, the undergoes modern packaging, but technology which is consumed advanced technology are needed to preserve product quality and meet market standards for superfood, alternative food, and also to support precision agriculture. Efficient food supply chains support for the distribution of food and the output for closer to consumers. So the squad board on farms and innovation in the must harness its genetic process and ecosystem diversity. Increasing productivity must match with efficient and technically and other food process.
Mohon maaf, suaranya sepertinya jelek di online. Lemah sekali, tidak bisa terdengar. Maaf, suaranya sepertinya jelek di online. Lemah sekali, tidak bisa terdengar.
Nggak usah, nggak apa.
Ladies and gentlemen, before we go to the main session of this event, we will have a group photo session. Before we do so, please follow the participant online. We ask you to make sure that you have used the virtual background that provided by organizing committee. Please activate your cameras. And for on-site participants, please remain seated and the committee will capture this moment.
Nah, ini udah sih. Ini lagi aku foto.
For online participants, the organizing committee will capture from the first slide, 1, 2, 3. Second slide, 1, 2, 3.
Cuma dua kok. Cuma dua.
And the rest of the slide will be captured by the organizing committee. Thank you for your attention. Well, ladies and gentlemen, now allow me to introduce today's moderator for today's event, Dr. Dwinita Utama, MSc. She is a senior researcher and serves as the head of Research Center for Horticulture at the National Research and Innovation Agency, BRIN. Dr. Dwinita specializes in plant genomic results bioprospecting to create superior lines through precision, smart, and eco-friendly farming. She has been actively involved in various national and international research collaborations, and we are truly honored to have her guide us in our discussion session. Please welcome Dr. Dwinita Wikan Utami.
Okay, thank you, MC. Good afternoon, distinguished speakers, guests, and participants joining us both on-site at KKB Kusnoto and online via Zoom and YouTube streaming. Welcome to the 12th webinar Ruang focus group discussion, and today the theme is about the synergy of superfood, alternative food, and precision agriculture for sustainable food security, which is organized by Research Organization for Agriculture and Food, ROAF, under BRIN. Today's discussion is part of the Nutrifoodsync initiative, aiming to accelerate collaborative research and innovation in food system. Let me begin by expressing our gratitude to Dr. Puji Lestari, Chairman of ROAF BRIN, for opening remark, but sorry, that's not really clear because I need trouble in technique. And all participants for your commitment to addressing Indonesian food security challenges. So I will remind the objective and flow of the season this FGD seeks to first is the map synergize between superfood, alternative food, and precision agriculture technologies, covering land agriculture and aquaculture fisheries to strengthen the national food security. And explore the collaborative research concept with stakeholders from academia, industry, and also government. The third, to strengthen the innovation ecosystem for sustainable food solution. Today, we have 4 esteemed speakers who will share insight in 15 minutes, around 50 minutes for a presentation, followed by maybe around 40 minutes, the interactive discussion. I encourage all participants to engage actively. On-site attendees may raise their hands, and while online participants can submit questions via chat box or maybe raise hand. So the first speaker, Professor Sastia Prama Putri from Osaka University. Our first session delved into the metabolomics approach for mapping food diversity in Indonesia. Metabolomics offer groundbreaking tools to unlock the nutritional potential of Indonesia's biodiversity. I now invite Prof. Sastia to the stage. Professor, you have the floor. Yes, please, Prof. Sastia.
Okay, very good afternoon. Assalamualaikum warahmatullahi wabarakatuh, rekan-rekan semuanya. Terima kasih banyak untuk kesempatannya hari ini. Very good afternoon, Dr. Dwinita. I hope you can hear my voice clearly. Can you hear us? Can you hear me?
Very
Yes,
clear.
it's clear.
The sound is not really, maybe please to,
I think the participants in Zoom can hear me clearly, but maybe the participants offline needs, it's okay? So please let me know whenever I can start. In Zoom, it's clear. Yeah, I think in Zoom, it's clear. So I think the volume in the offline maybe can improve more. Okay, please wait. Okay. Yeah, so I'm very delighted to be here with you today, although still via Zoom. And I'm really honored again to be given the opportunity to share about my research in metabolomics, which I think would be one of the key solutions for everyone who are interested in mapping our national treasure, which is in our biodiversity. So today I would like to emphasize 2 points, which I think would be of interest to everyone working on food and agriculture. Number one, how we could maximize the potential of our bioresources, especially maximizing the function to provide sustainable sources of food with relation to food security issue. But at the same time, we have to remember that food can also be our medicine, and we don't want our medicine to be food, right? And so for that, we want to encourage everyone to promote and raise awareness to many different kinds of health benefits we could get in food. So just a little bit about me. So I'm leading a research laboratory called the Global Food and Agricultural Laboratory, which is very much related to ROAF BRIN. And my research is dealing with metabolomics, which is a comprehensive analysis of metabolites. It's a combination between bioscience, analytical chemistry, and informatics. And therefore, we could use this kind of technology for many different kinds of biomaterials. So today, the biomaterial of choice is going to be food and agricultural product. And it's very useful for quality improvement, to assist breeders, to develop new cultivars, for trait optimization, and also for post-harvest management. And of course, when we talk about food metabolomics, it's very important that we address the social issues, especially the social development goals related to SDGs. So food metabolomics can be very useful to increase the shelf life. It's very good to improve the quality, authenticity assessment, diet monitoring, to identify unique characteristic in a particular bioresource, and therefore we could maximize the potential use of our bioresources, which will lead to product development. And I think it's very much in line with 3 different global issues, such as related to food security, improving nutrition, and sustainable agriculture. But also, I'm now very much interested to study about functional food, which ensures healthy lives and promote well-being for all ages. Under this collaboration, I have 2 main research areas. In the past, I work a lot with microbial strain improvement, where we examine the metabolism of microbes and how we could utilize this kind of information to improve the strain performance. And then I applied the same principle in microbe into food and agricultural product, where we emphasize on tropical foods, especially the foods in Indonesia. So metabolomic for strain improvement, usually rational selection is based on genetic knowledge. So when we do plant breeding, for example, we do a crossover and we select the best, and it takes time because sometimes it might result in unexpected phenotype. So metabolomics is the final readouts of biological system, and it is very valuable to provide us with deeper understanding of the metabolic state of host strains, which will then facilitate strain improvement. So under the same scope and the same framework of thinking, we think that understanding the metabolome many different kinds of metabolite that is involved in metabolism is very much correlated to the phenotypic features, which then contributes to the final phenotype as it is an end result of gene expression. And therefore, we would have deeper understanding of the metabolic state of the host strain. So when we talk about many different kinds of microbes, our research have already demonstrated a very powerful technology of metabolomics to improve strain performance. And with improving strain performance, that kind of technology and kind of framework, we could translate it to improving food quality. And we could also use it to improve food quality in terms of its nutrition, in terms of its sensory properties, the taste, the aroma, or we could also improve the health benefits of food, which use the same principle. So then I started working on metabolomics to apply this kind of technology by doing the analysis of comprehensive profiling of many different small molecules involved in metabolism, analyze them against many different kind of phenotypes, and apply them to many different kinds of food and agricultural products of Indonesia. And I would like to show just one of the research that we have been doing with coffee, which could encompass the study about authentication, improving the quality, geographical origin, and also improving the altitude. So, for example, if we understand what makes coffee delicious, and we could make a correlation between coffee quality and metabolome profile, we could then identify markers that are important for coffee quality. Then we could go back to the breeders, we could go back to the post-harvest researchers to inform them strategically. If you want to improve coffee quality, then these are the markers to improve. Because if you improve these markers, then it would translate to improving coffee quality. Then we could then understand what makes these markers increase. What are the factors? What are the conditions in the environments? What are the processes that we might be able to investigate based on this kind of issue? So for example, there are many different kinds of steps in producing a very good quality coffee. And if we could understand the effect of many different kinds of influential, whether it's external or it's internal, we could then go back to the process, optimize the process further, and finally, by the iteration system, by building appropriate system, we could analyze the coffee, improving the coffee, analyze them further, and this kind of iteration, we could result in series of cycle of improvement of coffee quality. So this is what we have been doing for the finding the quality related metabolites in Arabica green coffee beans. So we use trained panelists. We analyze the coffee quality quantitatively using a trained panelist is a Q grader. We use it for 10 different samples from all over Indonesia having different cupping score. And then afterwards, we apply this technology by doing an extraction. And this is the way we do the extraction. We input the data and extract to GCMS metabolome. And then afterwards, we build a prediction model where we could find the correlation between X variable, which is the metabolome information, and the Y variable, which is the quantitative phenotype that we want to analyze. The quantitative phenotype can be coffee quality, can be particular nutritional quality, and also the same principle can be applied to particular health benefits. So you could use the Y variable. For example, the phenotype is antioxidant, anti-cancer. So you can use the Y variable as the anti-cancer activity, and it has to be measured quantitatively. So then you have this exploratory variable where you analyze many different kinds of metabolome, and you make a prediction model where you can find a correlation between the metabolite and the phenotype under study. Using this kind of principle, you could then find the positive correlation between metabolites with the phenotype that you are studying. It could be positive correlation or negative correlation, which also translates to what makes the quality of coffee increase as indicated by the cupping score, and what kind of metabolites are negatively correlated, which translate to compounds that probably are not good in coffee. Maybe the compound that makes it too astringent or the compound that spoils the taste of coffee. These are probably the compounds that negatively contribute. And then you go back to the process, feed this information into the system, to the on-farm breeders, to the post-harvest researchers, to the people who are working in the technological process of food. And you tell them, these are the metabolites that are positively correlated. These are the metabolites that are negatively correlated. What can we do in our system, in our processes, in our way to produce this coffee so that we could design and strategize systematically on how we could improve the quality of coffee by targeting these marker metabolites. So this is what we did. We have the metabolites that are positively correlated, which is metabolites that are positively influencing the quality of coffee, and metabolites that are negatively correlating with the quality of coffee. And these metabolites, we could understand what are the functions, and then we go back and target these metabolites, and we prove them by analyzing 35 different samples. We validated our finding and test more metabolites, more samples, and see in different samples whether the same marker metabolites also still show correlation to the coffee quality. And using this kind of study, we found that galactinol is a quality-related metabolite to coffee quality. So we could identify for the first time a new marker for coffee quality, and this could be the target metabolite that we could use to improve breeds, cultivars, post-harvest processes that would contribute to the increase of galactinol. If we can then increase galactinol, we could then think that we could improve coffee quality. So this is the framework of thinking. And using this kind of principle, we have applied the same technology to many different kinds of food in Indonesia, also in collaboration with ROAF BRIN. And this is one example that I did to improve the health function of tempeh. So how we could improve our diversity of food and reveal hidden function in food that we normally eat, but we don't know what is inside our food. So this is another case study using food as a sample. Perhaps everyone here is very familiar with tempeh, but mostly probably you know only tempeh contains very good compounds like antioxidants, isoflavones. And you also know tempeh is good for cardiovascular health, but actually tempeh can offer much more than that. So with the food metabolome profiling, we were able to expand our knowledge of what is in tempeh, and we could improve our strategy by analyzing more compounds in tempeh. It's not only focus on one or two compounds, but we could combine many different kinds of analytical instruments to improve our knowledge on what is in tempeh. So what I did was I analyzed using GCMS, and we could analyze small hydrophilic metabolites. I annotated 74 metabolites. We also use untargeted LCMS. We could analyze maybe more variety of metabolites related to secondary metabolites. And we analyze also the isoflavones. We target 12 different isoflavones. Using this, we could analyze a total of 204 metabolites from tempeh. We subjected these compounds into different kinds of database like PubChem, DrugBank, and CHBI. And then we could identify most of them, the ones that are important for tempeh health function. These are the isoflavones. But we found a very interesting compound called meglutol. And this meglutol has been reported before to be very important for reducing LDL cholesterol level. So these food metabolome profiling in correlation with blood plasma metabolome profiling in Harvard Medical School, we found that meglutol in the blood plasma, the higher the meglutol concentration in blood plasma, the lower the LDL cholesterol. So we found in more than 1,600 participants through the data that plasma meglutol is inversely or negatively correlated with LDL cholesterol. So this kind of framework can also be applied to Indonesia, where we could expand our food metabolome library, and then we could make a correlation with the human plasma metabolite library, connected with the possible health outcome, in this case, LDL cholesterol, but it could be any other things like cancer. It could be the risk of lowering diabetes and etc. And then we could probably find a new functional component in Indonesian local food. So this is one of the demonstration where we could show that tempe is a very superior source of mangglutol. And we think that in the future, we have very bright future because now we have facilities in BRIN that could analyze many different kinds of metabolites, and we could apply the same framework, the same principle by expanding our knowledge on what is in the metabolome of food and make a correlation between the human metabolome data and the health outcome. So using this kind of framework, number one, we could improve the food quality and any quantitative phenotype that we would like to target. And number two, we could explore hidden health benefits in our local food to develop new functional food, new functional component that might be very valuable to improving the health benefits and involving both issues on sustainable source of food, which is related to food security, and also to improve the health benefits of people, which is also a very important issue in Indonesia to reduce the occurrence of very serious chronic diseases. So with that, I would like to thank everyone for the opportunity to share about my study. And I would like to thank my collaborators in Harvard University, as well as my mentor, Professor Eiichiro Fukusaki, and all laboratory lab members that are involved in it. Thank you very much.
Okay, thank you very much, Prof. Sasya, for highlighting how metabolic can guide superfood development. Thank you very much. So, due to the Prof. Sasya have any agenda, so the discussion and maybe if there are any question for Prof. Sasya, just please, because Prof. Sasya have another agenda. So, any question or Prof. Sasya? Is there also in here, right? Did you want to asking some? Is there?
There's somebody in Zoom.
Okay. Hatim Al-Basri, please.
Yeah, thank you. Chair. Sorry, I can't open my camera for some reason, so hopefully that is okay. Thank you, Prof. Sasya, for your very interesting presentation. I think this is a new work for me, and then I think it's a really a nice research result that you have presented before just a few minutes ago. So, but from your presentation, I think based on my really, what do you call it? Really shallow, for my really shallow understanding of metabolomics, you presented the goodness of metabolomics, but I haven't seen any, let's say, risks from metabolomic studies. I think that would be great if you can explain also, are there any risks that comes with the metabolomic studies or metabolomic, approach, which I think us as a what do you call it, a really not that person involved in that kind of thing, also understand the potential repercussion of that kind. Yeah.
Okay, thank you very much. I think it's a very clear question. So there is no risk in doing metabolomics actually, because it's just analyzing. It's like a food analysis. So if you want to analyze like the composition of sugar, amino acids, is a method to analyze, but we analyze it more comprehensively. It's not, for example, just analyzing the protein content, but instead we analyze the amino acids and the organic acids. So there's no particular risk, because what we're doing is that we're not changing the the structure, or we're not modulating the genotype of the food itself. So for example, if we do GMO, we are altering the genetic sequence, right? But with metabolomics, what we are targeting is we analyze what is in our coffee, for example. Then we understand what are the importance of particular compound in coffee. So then what happens afterwards is how we can improve this coffee quality. If we could choose methods that are not dangerous or are not risky. For example, how to brew the coffee, how to do the proper post-harvest. So what you're doing is modulating the final phenotype. It's not necessarily changing the plant material. So because the metabolome is the end product, right? It is an effect of many different factors, including where we grow the coffee, how we produce the coffee, how we do the post-harvest, how we could roast the coffee, how we could do. So I think that would be a very yeah.
Yeah, can I just follow up from your description?
Yep.
Yeah, so yeah, I think in the study, of course, there'll be no like changes on the content of the food itself. But I think you mentioned that based on your study, and then you inform the breeders, the farmers, if you want to have this kind of level of, for example, the glutenol, you have to add something, which raise a question, if a particular metabolite is increased, is it gonna change the whole metabolite, a system or something like that, which probably can result in unwanted, result or something like that? Thanks.
Okay, I understand what you mean. Yes, that is a possibility. So if, for example, we want to increase the concentration of meglutol from tempeh, let's say, yeah? So there's always trade-off relationship because the metabolome, input and output, right? In the metabolism, if we alter the metabolic route, for example, now it makes more meglutol, there's a possibility that other compounds that use the same substrate with meglutol biosynthesis, the other compounds will be decreased. So there's a possibility, for example, with the increase of meglutol, there will also result in the decrease of other metabolite. So it's a trade-off relationship. So something we want to increase and something it could decrease. But that is not the goal of metabolomics. The goal of metabolomics is to give the information that we could have high meglutol and what is correlated. Metabolomics is up until there. Then the strategy, how you use metabolomics is up to you. So it's already outside of metabolomics. So metabolomics it's a hypothesis generator. It helps with building strategy for strain performance and strain optimization. But that is a follow-up experiment after you do metabolomics. So then if you would like to use metabolome information to improve a particular trait or a particular strain performance, then you need to consider, okay, for example, I want to increase meglutol. What is important phenotype for me? Is it only the meglutol? Or I also want to improve the taste? Because there's a possibility when you improve the health benefit, you decrease the taste because then it becomes bitter. For example, like you want to increase epicatechin in tea. Epicatechin is very good antioxidant, but also it tastes bitter, right? So then you can choose which one is more important, the health benefit or the bitter taste. So metabolism is metabolism in food and plant, it's intercorrelated. Something increase, another thing decrease. One phenotype improve, there's a possibility that another phenotype decline. So that would also be a part of the strategy for the performance, then we have to decide. I hope that is clear enough, Ibu Dwinita.
Yeah, I think
What happens after metabolomics
Yes.
is very important. Yeah. Okay.
Yeah, I think it's clear already, right? So that's why metabolomics is only one a part of the characterization actually. So
That's right.
the strategy is which one, how we can know a lot about the pathway, something like that. So that's why we can priority which one the enzyme and like this. So this one is no really something like that because this was already inside in plant, right? In this cell, I think. So I think it's clear. Pak, is there any? Please, maybe. Yeah. Here, I think.
One more question, ya Bu.
Sure. Yeah. Yes. Thank you. Thank you very much. It's just the one in here.
Thank you so much. Very interesting presentation. I have like maybe 2 or 3 questions, but then short one.
Just one, please. One, please.
Just one.
One question.
Yeah, I just like want to know about the tempeh. You find like about more than 100 metabolites. It is from GCMS or LCMS only, or together from these 2 analyses?
Yeah, I used 3 different analytical instrumentation. In total, 204 metabolites. That is annotated. So I used GCMS, LCMS, and targeted LCPDA for isoflavones.
Okay. So that's the first one. Maybe just like another short question. So,
Yeah, sure.
Yes. Me and my team actually did in vitro culture of Indonesian ornamental plants. And fortunately, we find very interesting metabolite from this callus culture. And this metabolite is quite common use for the beauty cell, for the beauty ingredients. But then we don't know how next step, because we know how to produce it, but then we don't know how to, to get these metabolites from that callus, and then what to do next, basically. Can you give us the suggestions?
Okay, so first of all, thank you so much. And I think it's very beneficial if you could design many different kinds of process conditions. Design the experiment, for example, production A, production 2, different condition means 3 different methods to produce callus. And then analyze the metabolome. And then you find that this particular condition translates to this kind of metabolome profile. And using this kind of information, maybe then you could you could do the next experiment and understand the influence one by one. Maybe based on the different condition, you can make a correlation with certain concentration of metabolite. And then afterwards, the next experiment, you could maybe analyze another condition. Then afterwards, you could do a strategic optimization of the culture condition. So it's not like trial and error, but it's guided by the knowledge of metabolome. Maybe that is one thing that I could advise to start.
Yeah, thank you very much, Prof. Sastia. It's very nice, your presentation. And yeah, bye-bye. See you. Thank you. Yes.
Thank you so much, everyone. Thank you.
Thank you. Okay, we continue our session. I think we have the second speaker, the Professor Mahmud Iwan Solihin from UCSI University. So in this session, we shift to technology with deep learning and computer vision for precision agriculture. Prof. Mahmud, please share your insight on AI-driven solution for farming efficiency. Yes, please, Prof.
Oke, thank you. Terima kasih, Bu Dwinita. Screen sudah terlihat ya?
Sudah, Pak.
Oke, terima kasih atas undangannya dari ORPP or ROAF. Oke, right. Terima kasih Pak Yudhistira, Pak Destika, yang menyampaikan undangan ini. Oke, saya akan lanjutkan dalam bahasa Inggris ya. Oke, so I introduce myself. I'm Mahmud Iwan Solihin I'm neither from agriculture or food research actually background, but I like to collaborate with different disciplines actually through AI, especially recently. Right, let me see. Let us see that hopefully this my talk will be beneficial for us in terms of building collaborative research, right? Okay, so myself actually I'm studying from Gajah Mada University, S1 Gajah Mada University, electrical engineering, and I continue study master and PhD in Malaysia International Islamic University, and currently I'm in mechatronics department, right, where I originally engage with control system, robotics, automation, and then goes through with AI optimization, and I have done also some research in spectroscopy, actually the chemometric, so-called intelligent chemometrics, and lately after 2020, I also go deeper into deep learning computer vision, right? So that's actually a journey of research of that my opinion is or my vision is that I want to have a more wider spectrum in terms of the knowledge and collaborations, right? So I hope not to be shallow for this presentation, for especially for those who are learning AI, right? So what is computer visions? And I'll bring to the more like general overview in the context of food and agriculture. Right. I think everyone knows that AI is basically trying to mimic the intelligence of human beings, right? In terms of the thinking skill, in terms of the decision making, right? And computer vision is in terms of the vision of the related to the seeing the world through the image and videos, right? That's basically computer vision. Therefore, AI scientists develop model that can have the capacity of thinking as humans see the world, right? So obviously, if we talk about computer vision, we'll talk more into the data in terms of image, right, and video, right? And then the definition of precision agriculture, I think everyone's like knows already, right? In this forum, I think that basically, again, it is interdisciplinary research. Okay, it involves some kind of different background to go deeper, right? So that we can involve AI, we can involve GIS, okay, and then of course the agriculture knowledge itself, okay, to make decisions. And then I'm going to introduce the basic of fundamental of computer vision actually. In as far as now we're concerned, actually there are at least 4 application or subfield in computer vision to do the tasks. Okay, so the first one is of course image classification. That is basically simply to distinguish between different type of image in an image, right? So for example, here we have cat, okay, that human able to see, right? Is by pointer seen? Okay, like this, right? So that's image classification, and actually the application is in at least in the research is quite a lot in many field of application, including agriculture. Okay, and then we have object detection. Okay, that is actually we classify as well as locate the object in the image. That is localizations, right? So that's actually object detections. So we here we have dog, we have cat, and that is the location, right? The given bounding box here. That's in the training process of the model, AI model, deep learning model. Actually, the human need to do annotation, right? That is the drawing the image. So if we have many data, that means the annotation job will be very huge, right? It will be very troublesome and taking a lot of time. And then we have also instance segmentation. That is like here, the boundary is not bounding box, but it is like pixel to pixel boundary here. Like this is the animals. This is the cat, and here the different cat here, two cat here, red and yellow, and the background is not segmentized, right? So that's basically segmentation, instance segmentation, and then we have semantic segmentation that is also include the background, like this is sky, this is the grass here. That's another type of segmentation, right? And I think if we talk about the nowadays common research in machine learning, deep learning, we have at least two very famous applications. One is regression. I'm not talking just now. I think the Dr. Sastia presented about regression that we can do also with machine learning, right? And if we talk about classification, I think we will have this kind of metrics to see the accuracy, so-called confusion metrics, right? If the control is positive, right, say the plant has disease or not disease, then okay, this positive, and then prediction is positive, of course it is true, positive. Okay, if it is really negative, like during the COVID, we have, I think, negative COVID, and then prediction through PCR is negative, that will be true negative, right? But I think last time I'm observing that COVID case has many false positive, okay, to make healthy people become like positive things, okay? And in my opinion, it is too much false positive in the COVID estimation through PCR, I think, right? But then in the object detection, how I can this? Okay. In the object detection, the metric is more complicated. That if, let's say, the ground truth is the red one that is being annotated by humans, right, by expert, and then the prediction is red, and then it has certain level of overlap, that is true positive, that is correct prediction, right? But then if it is, let's say, the bounding point is correct location, but then the classification, the class is here, bird is being set as or being predicted as cat, that is wrong, right? Then it is false positive, right? And then if we have bird here, and then only predicted by the small overlap less than IOU interaction of unions less than half, right? This overlap is less than half, that's become false positive. And here, like no overlap at all, of course, false positive. Here, no prediction, then come false negative. And that is the derivation of the terms in object detection, right? And then the threshold usually we use in the object detection model, we use, let's say, 50%, and then also 50% to 95%, and then being average, and that's called the mean average precision, right? What's next? Okay. So I think because I'm going to go to the some kind of trends later on, then I think the advantage of deep learning is that the feature extraction process is performed automatically than using the machine learning, okay? And the strength of machine learning is basically when the input is tabular instead of image, right? When we talk about deep learning, beside the generative AI things, actually the input is image, okay? And the extraction process or the feature extraction process is being automated in the deep learning algorithms, right? So the strength of the machine learning is on the tabular data, right? The strength of the currently deep learning and computer vision is on the image data, right? This is, I'm not talking about the large language model, about the generative AI, right? That's use the language input, right? That is also deep learning, but not in the context of computer vision, right? Okay. Next. So the key advance in computer vision is, I think, the big data availability, right? Annotations, and also the computational GPU power, right? And also the power of pre-trained model that is being, like, the model has been trained through a huge number of images collected by big companies like Google, like Microsoft, and then the model is being adopted into different or custom data set that we use for our purpose, especially or for example in the agricultural application, right? Okay, and as I said just now, like for example, this is the common so-called COCO dataset. This common object, common context dataset by Microsoft. Like this kind of image containing, if not mistaken, 80 object categories, like person, dog, so on. Okay, that's being labeled by the team actually. So you can imagine labeling this 330,000+ image, okay, manually. So this is example of annotation tools, right? The label me. So we have to create this box and then name it like bicycle, dog, and so on. Okay, that's annotation is taking a lot of time, right? And then I'm talking about the recent state-of-the-art deep learning and computer visions. As you see, actually, the most classical and popular pioneer of deep learning is CNN, convolutional neural networks. Okay, all these are actually including VGG, GoogleNet. I think this developed by a company or a group of researchers, right? Starting from especially after 2010 up to recently before 2020, right? This is for image classification tasks. Okay, we have some for object detection tasks, for segmentation tasks, and so on. And if you look at this, especially for the later one, like after 2020, 21, 22 here, you see here, we have the transformer base, right? Like DETR, okay, like here a swing transformer. Okay, that is actually derived from the transformer, include the transformer model that is also used in the like GPT, like the large language model for the generative AI, basically. So it's like trying to adopt, which is the transformer, which is originally used in language research, NLP, the subfield of AI, into the computer vision, right? So I think this is something in the area of deep learning, something that we can explore, right? The application of this transformer-based either classification or object detection or segmentation model, right? And this is, I think, I saw as a trend of research when we go into the deep learning computer vision research. Okay, and then now in precision agriculture, I think as I check on the publication review, okay, at least there are main application, okay, in the crop management, and then harvesting management, soil management, water management, right? And most of the publication, I think next we can see in the crop management, I think. Okay, and then less in this is published in 2025, a paper I read just by chance. This is from Malaysia University publishing this paper, right? And this is review from after being screened with the method or Prisma method in review paper. They publish, I mean, they select 200+ papers, and they conclude this as that crop management dominating, okay, the population with this kind of subfield of disease detection, weed detection, crop detection, plant nutrient detection, growth monitoring, phenotyping, and yield estimation, right? So I think crop estimation and disease detection are among the bigger proportion. Okay, and also we see this in terms of the source, right? The data source imaging. We see here, of course, the RGB image normal camera is still dominating, right? The blue one, and second one, I think, multi-spectra, right? The orange color one, right? Remote sensing also there, but not as much as RGB. I think it is interesting if you can see this, like how the data is collected, for example, using UAV, for example, how and so on, I think, but I do not see in the paper. Okay. And then now next, looking at the, because last time I'm writing a proposal with one of the member from ORPP, right? And I'm seeing this review. that actually the trend is in the data fusions, right? This is also paper written in 2025, right, on remote sensing data fusion for agriculture application. And we can see here the data fusion, remote sensing, UAV are actually among the outstanding keywords here, right, in the research. And if we talk about multimodal, as I said before, that machine learning, okay, like neural networks, multi-layer perceptron is with the strength in the tabular data. Like for example, we have agronomical data, weather data, and so on, tabular, right? Soil data, right? And the strength is being processed by in the machine learning. And then we have also image data from the plant level, or maybe from remote sensing, or from other types of data source, that is image, and then we put into the deep learning. So this is like combining the capacity of machine learning and deep learning, and then we combine in the feature fusion here, right? That is feature fusions to make the final decision, right? So for example, here, I think from this paper is on the, let's say, rice disease diagnosis. Okay. On the pattern search, I also try to look at this keyword, deep learning, computer vision, agriculture. So the trend from 2020, especially, we have growing large numbers, right? And it's still increasing until this year, 2025 is this, right? Okay, and I think 2026 is coming. So the domination is from United States, and I think this World IP organization may be involving multiple countries. And I do not see the thing is that from much from the Asian countries. So I think this is opportunity to explore, like with the BRIN Center, right, on this field as well. Right, Malaysia, Thailand, no such thing. Singapore even maybe under WIPO here. Yes, right. And lastly, about the challenge on applying the AI-based or deep learning-based computer vision on this field, I think is first of all is the availability of the annotated data or grown truth things, right? So of course, to train the supervised learning, we need a reliable ground truth, right, which is very difficult sometimes to collect through the human, right, worker, right, expert, right. Sometimes also we see that negative sample are very less. Like for example, healthy plant is of course dominating the unhealthy plants, right? So that's creating class imbalance, and we have to deal with that, right? The model has to deal with that. We have also the challenge like seasonal and regional variability, right, in terms of spatial and temporal variability. And we have also environmental factor like weather, lighting, cloud shadow. I think in remote sensing, we have this issue also, occlusion, right? So, the object overlap with one another. Okay, and then we have the balancing between accuracy and complexity, right? When we deep learning researcher trying to have accurate model, then the complexity increase, the model is become heavier, it's become like heavy to be put in the edge device into the deployment, right, into the application, right? So that's the issue is coming, like the issue of making a lightweight model without sacrificing the accuracy, right? And then we have also the challenge on integration of different type of data, okay, from different type of source in the multimodal approach, right? That's the problem of integrating different maybe spatial resolution. We have different temporal resolution and so on. And lastly, I share about my experience in this area. Actually, I have collaborated with applied science earlier, mostly in with the intelligent chemometrics. The last time we used this handheld spectrometer, right? It's just like a hand palm size, right? And we have managed publishing this, like for example, to have research in honey adulteration detection. We have milk powder. Okay, and then we have some review paper. And recently, this is my student is publishing in what's called compression, right, to make the lightweight model just now, right? And I think this is last slide. So we can explore some potential joint research, especially with not only actually remote sensing. I think in the ORPP, I think you have the expertise also in remote sensing. Okay, so maybe we can collaborate with these topics. Last time I have submitted one proposal with one of the member in ORPP, right? And this is the topic about the water stress mapping, okay, for plantation with remote sensing. We can have also potentially like maybe estimation of carbon stock, right? And we can have also non-destructive food fraud detection. We can also work in intelligence chemometric like the one I showed just now. Okay, that's all. Thank you, moderator. I returned
Okay.
the times.
Yep. Thank you very much, Dr. Mahmud, for that groundbreaking perspective, I think, on how artificial intelligence and also computer vision are revolutionizing proper precision agriculture. So, may I let's next presenter. So the third presenter is Pak Yudis, Dr. Yudhistira Nugraha, who will be discussing about the optimizing the production through precision agriculture. Yes, Dr. Yudhistira, over to you.
Okay. Okay. This is already slide show, I think. Not yet. Wait for a second. I have a trouble with with the device. Okay. Enggak ya? Oh ya, it's okay.
Maybe just the percentage is more bigger, I think. It's okay, right? Sorry, is there any problem in here?
Sudah bisa ya? Iya, nggak apa-apa.
Maybe at the percentage, I think. If is it not to slideshow?
Tapi nggak bisa slideshow ya?
Is it the problem for slideshow?
I will transfer the PPT to.
Excuse me, please wait a moment.
The slide is showing onlinely.
Yeah, but it's cannot do a slideshow to make it bigger. Is it fine in the Zoom? Please?
But cannot move.
Oh, cannot move? Oh, that's maybe the connection or what?
I already transferred.
Yeah, please wait a moment.
Try to stop it again and and try to reconnect it.
Okay. Bisa?
Start first. You
Okay,
can
okay,
start
okay.
and
I will
then-
start.
Yeah.
Okay, thank you very much, the moderator. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. The Honorable Chairman of Research Organization for Agriculture and Food, Ibu Puji Lestari, and also here we have head of the Research Center for Estate Crop, Research Center for Animal Husbandry, and also the distinguished speaker from our collaborator from international and national, and also we here have Puji Lestari, and also here we have head of the Research Center for Estate Crop, Research Center for Animal Husbandry, and also the distinguished speaker from our collaborator from international and national, and also we here have Puji Lestari, and also here we have head of the Research Center for Estate Crop, Research Center for Animal Husbandry, and also the distinguished speaker from our collaborator from international and national, and also we here have Puji Lestari, and also here we have head of the Research Center for Estate Crop, Research Center for Animal Husbandry, and also the distinguished speaker from our collaborator from international and national, and also we here have Puji Lestari, and also here we have masuk Zoom. Tes. Halo. Okay. I'm sorry for the trouble. Okay, I will start my presentation. Last two presentation is very detailed, right? It is about the metabolomics technology in agriculture and food, and also the precision of agriculture. So I will talk about the new platform that will facilitate the collaboration between any institution, national and international. So this new platform before the new platform is already agreed by our chairman, the chairman of the BRIN, and the deputy of the facilitation of research in innovation, that this platform will be started in next year, in 2026. So this platform is optimization of agricultural production and the development of agricultural product. So, and the deputy of the facilitation of research in. Oh, ada rekamannya. Atau di sini aja. Udah. Oke. Di situ kelihatan ya. Oke. Oke. So I can talk with the device. Okay. So sometimes technology make us difficult. So nggak apa-apa kan? So in this slide we saw that technology is very important for leveraging the agricultural growth. So there are three factors. There are three factors that affecting the agricultural growth. The factor that can be independently influenced by the humans. So it's government policy, investment of agriculture, and we will talk about the technology change. So in this session, we talk about how to increasing the human capacity resource and also regarding to the research and innovation. So this without slideshow is okay, but
the percentage should be at more.
Yeah, okay. The point is, the innovation is very important. Without innovation, we can have a growing the productivity. So, in the case of the rice, in last decade, we have no growth of the productivity of rice, our rice production. So some social economists say there is slow down the adoption of innovation. So if the innovation can be implemented to our farmer or our, in the case of the rice system, there will be increasing the productivity. In last decade, we have no growth of the productivity of rice, in the rice production. So innovation for growing productivity is using the precision of agriculture. The adoption so using the precision of agriculture, if the innovation not only can be implemented, make the productivity increase, and also it will have we can save the inputs. So agriculture inputs such as fertilizer, pesticide, and innovation water. But also productivity is using our environment because some the chemical product can be hazardous for our environment. So regarding to the precision of agriculture, it's already mentioned by Professor Iwan, very detailed, but I will repeat again. The precision agriculture is like the core of the digital agriculture and smart agriculture. Digital agriculture, we talk about real time. It's about the IoT, robotic, sensor, and drone. So while the smart agriculture is talk about the connectivity, it's talk about IoT, AI, big data, both real time and connectivity. It is cannot be said that precise, but the precise agriculture is talk about the optimization. It's about the spatial temporal modeling, remote sensing, big data, communication, and other things that make digital agriculture and smart agriculture more precise. So, ini dapatnya ya, nggak bisa ini ya. Okay. Next, ini bisa ya, tapi belum ini. Okay, it's okay. So I think Pak Mahmud, Professor Mahmud Iwan has already explained more detail, but I will repeat again. So the precision agriculture is talk about information-based management system. It's about how to capture the phenotypical of object. It can be crop, it can be fish, or it can be livestock. And the captured object can be processed by the computer or software. It will generate the data, and then the data will be used to create an artificial intelligence, and then in artificial intelligence can be implemented for the actual or the acquisition of the object. For example, for make a optimum of dosage of the fertilizer, for how to put insecticide and so on. So the platform can be used many tools. It's from the sophisticated tool like satellite or drone. It's also like we can optimum of dosage of the fertilizer for platform like how to digital camera or sensor. So in our research center, we have used a deep learning model to predict grain yield in rice. We have generated data from 105 rice accession and in 2 locations, and using the convolutional neural network, we can predict the prediction, and actual rice can be predicted as much as 61%. So it's quite precise. And this study is one of the example how we involved the student in this study. We have a student of degree by research for working this study. And also more advanced tool, we use a drone. We can capture the imagery of the rice field and predict the health of the rice plant. So we can decide how much the fertilizer is needed for make the optimal yield from this rice field. And also we can identify where the area that need to be put attention, for example, spraying of the pesticide or watering. So the more advanced is used by use a satellite. So our in BRIN, we develop a platform we call a GeoMimo, Geoinformatic Multi-Input Indonesia. These satellites can capture the imaginary, how much the rice field that can be harvested. So our government can decide the policy whether it is need to import of the rice or not. So it's kind of example for satellites. Tool is more what do you call it? It's more broader aspect. It's more general for government policy. So, but for drone and low cost phenotyping tool is for more specific or more detailed. So regarding to the precise agriculture, it's also including the breeding or precision breeding. In this precision breeding, we can involve many aspects like omic technology, for example, genomic, metabolomics, like Bu Sastia has mentioned before, and even it's panomic. So metabolomic, genomic can be what do you call it? Can be bridges for the breeder to make a good decision whether their material can be selected or the population or the individual targeted trait can be selected. It's panomic. So it can be also involved like another technology, for example, speed breeding. So in the speed breeding, we can use haploid technology, the double haploid technology, rapid generation advance, and also petoperiodism technology. So within this technology, we can accelerate the breeding cycle. So for conventional breeding, maybe we can take more than 5 times for rice, for example. Yeah. But using this technology, we can shorten the cycle of breeding within 2 or 3 years. So our platform will be our platform will be our activity will be about the precision agriculture for breeding and for the optimization of the production in the on-farm section. So in this platform, we also more optimizing the technology that we have developed previous in the recent year and the previous year, and we have also we open to collaboration with other stakeholders like in university, broad and also national university, and also we also have like to downstreaming the other platform in BRIN. We have like a structural biology platform. So it's characterization of the molecule, macromolecule and macromolecule. So within this platform, we can downstreaming this upstream research to make applicable in agriculture. So we have 4 themes. It's about research on optimization agricultural crop production, research on optimization livestock production, research on fishery production, and research on agricultural product development. So there is 4 themes that we can open for call proposal for other institutions, and also we open for collaboration. So we involve many stakeholders in this platform, and the goal of this platform is how to create technology, intellectual properties. Also, we also targeted some journal can be published in international journal. So we have also like research mobility, degree by research. We targeted 30 degree by research in 1 year. So we open for national institution or from the external of BRIN can joining this program, degree by research. And also we invite researchers from abroad as a visiting researcher. We targeted 5 visiting researchers. Maybe Bu Sastia and Pak Mahmud can be joining in this program. And also we open postdoctoral. Like here we have 2 postdoctoral attend in this session. So we have a research facility, genomic laboratorium, and also we have a greenhouse. We also have developed other facility, but it's under a provision, like a field experiment, precise field experiment, a plant phenomic facility, and also other thing like stem cell culture and etc. So I think this is my my last talk. I think with cross-sector strategy, we can implement the precision agriculture, including research institution, industry, government, and other things. And through this strategic invitation, can be initiated by BRIN. Thank you so much.
Okay, thank you, Pak Yudis, for outlining precision farming platform. So I think it's the, I'm sorry because there's any problem for the material in the slide PowerPoint. You can share in chat box the PPT. You can asking about the substances of the presentation. Okay, I think we find in the 4th speaker, the last speaker, Dr. Satrio Kridowahono. This is the final presentation focusing on innovation and superfood and alternative foods by Dr. Satrio Kridowahono.
Sorry,
Dr. Satrio, please proceed.
Minta maaf Bu Dunita, saya interrupt sebentar. Sebab saya 15 menit lagi saya harus keluar meeting ada di kelas. Kalau ada pertanyaan bisa saja sekarang, minta maaf.
Bu Nita, di open pertanyaan dulu saja buat
maybe
Prof. Mahmud.
The last session. The last session. So the discussion maybe after Pak Satrio give a presentation, right? Yeah.
Prof. Mahmud 15 menit lagi mau
saya keluar nanti. Kalau ada pertanyaan, kalau tidak ada nggak apa-apa. Saya keluar jam 4 Kuala Lumpur time. Okay.
So, is there any question for Professor Mahmud?
Okay, maybe Mr. Mahmud, please. If there is any question or discussion for presentation from Pak Mahmud, ada yang tanya, mangga hadir di sini. About the deep learning and also computer vision for the AI. Ya, Pak Markus. Yes.
Iwan.
Ya, Iwan.
Dr. Mak.
Ya, Pak.
Excuse me, Dr. Mahmud, can you hear me?
Ya. Lanjut.
Oh, okay. The presentation of the learning, computer vision, and agriculture precision. In Indonesia, we know that mostly, especially in horticultural crops, we have a multiple cropping or mixed cropping in the same area. The problem is how do we use the deep learning to disaggregate the complex of multiple cropping. Let's say within the same area, we have different types of crop, like we have chili, we have probably shallot, and we have lettuce. In terms of agricultural precision, how do we manage these three crops with different like nutrient requirement? Or probably they have a different disease infestation. So this is a real problem. Probably we can use by observing the images of the same area with different types of crops. Do you have any experience? How do we apply the precision agriculture within the multiple cropping area, especially in horticultural crops? The first question probably I can.
Okay, so very thank you. Appreciate the question. Very challenging question. Actually, that's basically also challenge to the machine vision. Actually, I give example not in horticultura. So, for example, in the project of tree counting, for example, for palm oil plantation, I think deep learning can do this already because we have the let's say using drone or using satellite data, we can count how many palm oil tree in the large plantation area. But then when it is mixed with the forest, like in the non-plantation forest nearby the forest, then the deep learning also confused in with the other tree. Like for example, of course the palm oil tree has similarity with the normal palm. Mungkin pohon palem biasa itu itu pun memang masih confusion. Jadi saya rasa sebab bagi perspektif saya I think cannot be too much surpassing the human capacity. If the human is still also difficult to distinguish, I guess the AI itself also still difficult to distinguish, right? Up to the moment at least. We don't know in the future of the progress of AI research and so on. Itu komentar saya, Pak. Jadi kalau untuk case horticultura tadi, saya rasa kompleksitinya mungkin masih sangat. We can try to distinguish that this is maybe a certain kind of tree, and then we can develop algorithm. This is another tree, and then we separate the segmentize the image like just now. So this classifying the image, so we can use a segmentation approach. But then I think there is still challenge on that difficulty to distinguish similar object. I think that's my point of view.
Yeah.
Thank you.
If any parent to validate the model if we build from the images interpretation, like if we want to see what the multi-covariate would affect special crop performance, probably we can say to distinguish crop with high yield and the other one with the low yield, and we can say this is associated with different, let's say, nutrient deficiency, the other one, and the other one about pest or infestation.
Itu ketitipan gitu. Kalau kita
Yes.
Okay, I don't know how to respond because the last few seconds there is noise.
Ini semua orang bisa mengerti bahasa Indonesia ya? Ya ini kita lihat Yang satu grup mengalami kekurangan unsur hara, kemudian grup yang lain ada penampakan di dalam citra. Kedua grup akan nampak di dalam citra sama-sama mempunyai simptoms yang nampak, tetapi satu disebabkan oleh nutrient deficiency, kemudian yang lainnya disebabkan misalnya tadi serangan hama atau penyakit. Nah ini kan dalam hubungannya dengan precision agriculture, mampu nggak kira-kira resolusi spasial berapa yang dibutuhkan supaya itu bisa diatasi, karena ini sangat berharga kalau bisa dipakai di level farm, level petani.
Oke, paham.
Seperti itu.
Oke, paham. Saya mungkin bisa kasih apa nih overview dari sisi apa nih sensornya ya. Jadi mungkin kalau kita explore misalkan image aja, kalau normal kamera warna dari sebuah objek itu sama, tetapi kita bisa misalkan pakai multi spektra kamera yang itu ada nggak cuma red, green, blue channel, ada infrared channelnya, ada even hyperspektra kamera yang dipakai di apa nih optical sensor. Nah, mungkin bisa memberi informasi termasuk chemical composition-nya gitu. Jadi saya rasa itu mungkin lebih kepada sensor development-nya. Tetapi kan mungkin kendalanya juga biaya ya, cost untuk implementation gitu. Multispektra kamera itu masih sangat mahal. Saya rasa kalau itu di apa misalkan di attach dalam drone untuk observasi lapangan itu mungkin saya rasa kendalanya adalah cost-nya. Itu mungkin komentar saya, Pak. Jadi mungkin ke pengembangan teknologi sensor untuk capture datanya.
Terima kasih Pak, maaf sebagian dicampur-campur bahasa Indonesia.
Ya. Ini kendala online dengan hybrid ini memang kayak gini, kadang-kadang kita yang online satu, yang offline yang lain susah. Terima kasih.
Ya, thank you Pak Iwan.
Ya, terima kasih. Minta maaf untuk Pak Satrio, next speaker saya ganggu sebentar ya. Saya keluar habis ini. Terima kasih.
Baik, terima kasih Pak. Thank you. Okay, still any discussion about the sensor? I think Pak Koko, you want to say something to sensor? No. Okay, we continue in the last speaker. Pak Satrio, is it okay? You clear the voice?
Yeah, testing for my voice. It's good.
Yes, so please Pak Satrio.
Okay. Thank you very much, Bu Dwinita. Okay. Hopefully there is no technical problem again for my presentation. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Honorable Chairman of Research Organization for Agriculture and Food of BRIN, Dr. Puji Lestari, all of BRIN College, Head of Research Center under ROAF, also all of research group in ROAF also attend this FGD. Also distinguished speaker, Prof. Sastia, Prof. Mahmud Iwan, Dr. Yudhistira, for very good presentation before my presentation today, and also all of the invitation for this FGD, International Focus Group discussion today in ROA variant number 12, particularly for our collaborator in this platform, in this in Superfood and Alternative Food Product platform, Professor Kim from PKNU, South Korea, and also Professor Chen from NTU, I see Professor Chen in the opening, but maybe now it's already leave from this event, and also Professor Peter from Fraunhofer, German, or maybe the team. I see there are some Germany person in this online meet and in this online event. This time I will present about the platform. We call it in Indonesia as RIM, RIIM 2025, particularly in strategic invitation, with the title Driving Innovation in Superfood and Alternative Food Product through Nutrifoodsync. If we talk about food, the background of this platform is related with, about global hunger, about food security, not only for Indonesia, but all over the world. We have the problem in food security. If we check the data at 2022, more than 700 million people suffer in hunger, and more than 2 and 4 billion people have insufficient access to nutritious food. So it's a problem for all of us. And for Indonesia, if we talk about food, we also have some problem related with how to food consume, some degenerative illness such as hypertension, cardiovascular, diabetic, anemia, and still has many problem which related with the consumption of food. In the other hand, Indonesia has so many potential of superfood and alternative food resources. Not only superfood, we have so many biodiversity which can proceed into the food in Indonesia because the variety of biodiversity in Indonesia. And if we talk about superfood, also some industry of superfood already growing up today. Some industries which produce particular product about superfood, it's grow and it's projected it will grow more and more in the further in the future. So I think we think that this topic, superfood and food alternative, and will be great if we can improve, we can give some additional technology to improve the superfood or alternative food resources, which particularly which come from Indonesian resources. That slide showing us about the biodiversity, and I already mentioned before that Indonesia has a lot of kind of commodities, a lot of kind of biodiversity that can proceed into the food product. Should be it can support food security in Indonesia, and also should be it has so many potential of food with uniqueness of nutrition and so on. So with this biodiversity, in here we not talk about the land biodiversity, but also ocean biodiversity. I think Indonesia has so many potential to improve for food resources. When we talk about biodiversity, it will be related. The first one about food alternative, as we know that the main component, the main part of food consumption is carbohydrate and protein, but the common people in Indonesia eat the carbohydrate, consume carbohydrate resources is rice. But actually, if we see this slide, there are 77 species of carbohydrate resources that can improve, that can utilize as carbohydrate resources. Maybe more than that, we can explore more. So it's possible to replace or to give additional, to substitute the common carbohydrate consumption of Indonesian people, with usually using rice, with another resources, but still carbohydrate. In the other hand, we also have so many species as the protein resources, not only from the animal, but also from the plant. We can see in this slide, we have 75 species of as the protein resources, which related with meat, egg, milk, fish also. And also we have 26 species of nuts, which probably provide us the protein resources. So we have so many alternative proteins, not only the common ones such as milk, egg, or milk, egg, meat, and fish. We can use another as the protein resources. In Indonesia, we also common consume tempe, as the Professor Satya mentioned before. Also tofu as part of another resources of protein based on plant-based. So we have so many resources to improve carbohydrate or protein alternative, which can produce and support the food security in Indonesia. This part about protein alternative. In another part, we also talk about superfood. In this slide, I have one of article from Fernandez Rios at 2022, one of the review of superfood in over the world. But the interesting thing in here, we can see there are so many kind of superfood which already mentioned in this review, but in this map there is no one commodities, no one resources of superfood which came from Indonesia. Actually, in Indonesia, if we see in this map, we can find cocoa, we can find maybe mangosteen, jackfruit, avocado, ginger, turmeric, and we have in Indonesia we have so many commodities which can involving in the superfood. That's the problem. We can identify from this map. Maybe the superfood resources from Indonesia not exposed much in scientific result, in scientific article. So that's why in scientific purpose in the future, we need to increase more article about superfood and also expose more the superfood resources from Indonesia. And actually in Indonesia, we can find some superfood product. The first one, we find one of the product of multigrain, which produced by some of industries. This multigrain contain many grain which characterized as superfood, which you can see in here, but still not much industries like this in Indonesia, but there are some of them already improved. Maybe in further it will be growth and there will be many industries will produce superfood, not only from the multigrain. In the right side, you can see tempe. Prof. Sastia already mentioned so many superiority of tempe. And in Indonesia, tempe is not only in industries, it's also part of SME. Maybe some household also produce tempe by their own. So it's already spread out in Indonesia, not only part of superfood, but it's already part of food alternative of protein resources. So actually, Indonesia already has a superfood in tempe. That's example in superfood. Now we try to see how improvement of example in food alternative. This one of our project at 2024 about rice analog. As I mentioned before, the common food of Indonesian people is rice. So if not eat rice, they said it doesn't eat. So if we have another resources of carbohydrate, we need to convert it into looks like rice or have a texture similar with rice, have a physical characteristic like rice, something like that. So we have analog rice project in here. And this one we still improve cassava analog rice as part of our project and still going in this year also. And for protein alternative, we have some research about in the left side, you can see meat analog. Meat analog, we improve meat analog not only from plant-based meat analog, we also improve meat analog from alternative protein. In this case, insect, grasshopper, which create as a meat analog, but have some obstacle related with the allergen. So we still improve the research how to reduce the allergen. In the right side, we also improve the meat alternative, not meat analog, but it's protein alternative, but not it's also correlated with tempe, but not from soybean. It's non-soybean. We improve some research correlated with the tempe from some local beans. You can see in here koro beans, pigeon beans, manding beans, peanut, even from soybean drag, which can convert into tempe. And we still try to characterize what is the superiority of each of these tempe. Not only about this, actually we have another example of research which still ongoing. Some of them related with the ocean resources. For example, microalga, spirulina, and plant micro or macroalga that also try to develop as the superfood. This platform, as we see in the previous background, the platform has so many benefits, but the key is it's explore local raw material, develop this material into alternative food or superfood, and then make innovative processing, introduce the technology, and make it as a product. Hopefully it has economic benefit also, not only we can get health benefit because of the superiority of food, but we can also get the economic benefit, social benefit, and as the technology, one of the goal is how to fill the gap from the previous review at 2022. So we have Indonesia include in the mapping of food superfood review article in the future. This platform, this project will be divided into three parts. The first part is exploration and development of superfood product. The second one, exploration and development of alternative food. The last one is the supporting technology for both of them. So if we talk about supporting technology, if we talk about how to preserve it and packaging, and how to make sure that it has assurance in food safety and the quality control, and also we think about the supply chain analysis in here, and maybe also in the how to overcome the waste, the food waste of from this product. This project will be conducted in 3 years. Hopefully can start at the end of 2025, or at least at the early 2026 until 2028. Yes, as I mentioned in the work frame before, it will be consist of research. The research area will consist of the how to explore, how to develop the product, how to make sure the preservative and package the particular preservative and packaging for each product, so it can keep the quality of superiority of the superfood, and also about the food safety. And the last one, if we want to make it as industries, we need to supply chain analysis to support this whole project. Also, it's not only about the research which involving the experienced researcher, but also have concern to increase to improve the researcher talent in Indonesia. So we involving some student with degree by research program of BRIN, also another program to support this project. You can see in the bottom part of this image, there are some target not only about the output, but how many researcher mobility, which involving the research talent of Indonesia that need to involve in this project. This is our scheme for mobility partnership, not only about degree by research, but also because it's international, we also involving a visiting researcher, visiting professor. So if you from abroad, from outside of Indonesia, want to collaborate this program, we have a scheme to facilitate your activity in Indonesia to support this project. This is how this project will be managed. The team, as principal investigator, me, and I will support by 3 young talent researcher here, Dr. Emma, Dr. Andri, and Dr. Nur Alim, with 3 different topic for this project. And until the day, we hopefully we can get about 15 billion Indonesian rupiah in 3 years, and we need collaborate with everyone of you which can support this project from the university, not on in the internal BRIN, also involving another research organization or research center, also from the international collaborator. As I mentioned before, we already have 3 or 4 involved. Prof. Sastia from Osaka also already joined to initiate this program. Also, we very open from industrial support to success to make this project successfully. To conduct this project, we support by BRIN with one of a big facility, food laboratory, with concentration in food. We have BRIN have a food laboratory specialist in Yogyakarta, located in Gunung Kidul, Yogyakarta, which provide instrument analysis, processing equipment for food, which concentrate in this laboratory. So if you want to join with this project, you will come to this laboratory as the main infrastructure for food research in BRIN. I think that's all of my presentation. Thank you very much for your attention. Research Center for Food Technology and Processing, Technology for Food, Food for Future. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Pak Satrio, for the comprehensive presentation actually on superfood and alternative food innovation. So I think it's all presenter is presented today, but I think we need also hear again about the program, and in here, Pubuji as the chairman of the ROAF is already here. So I think it's better if you say something about the examine of these two platform research. Yes, please, Pubuji.
Thank you, Nita. Good afternoon, everyone. I hope my voice is clear. I just would like to say hello to all of the partners joining online in this focus group discussion. Also, I would like to apologize that this morning my recording video for the speech was not so clearly, so I would like to explain a bit about what is the purpose of this FGD, with 4 speakers and 2 from other countries. One is from Osaka University, Professor Sastia, and the second is Professor Mahmud from Malaysia, and 2 are from BRIN. Yeah, Dr. Satrio Kriwahono and Dr. Yudhistira. Basically, we would like to socialize what kind of platform that we have, and we would like to engage with you all to be collaborative. How we could achieve the output, how we could get our proposal from the 2 platforms. So, as explained by the 2 speakers, especially from BRIN, so we combine in 2 themes, platform into 1 theme, the synergy of superfood, alternative food, and precision agriculture. So with this theme, I hope the mandate and task or function of your institution could contribute in this research. The way how to contribute is, because it is a kind of, I don't know how to say, it can be contributed in kind or support, because the PI will be from Indonesia, from BRIN, but you can support from your own, or you can share knowledge, or we can do together, start from beginning this research, you can be also expertise, and also knowledge transfer technology. And for the proposal can be composed between Indonesia and your site. So maybe after this FGD, each head of the center, each the PIC of this platform could communicate with you to develop a proposal of the research project. And the second is actually also, we are very happy that you could support our researcher mobility. For example, kind of postdoctoral fellow, visiting researcher, visiting assistant, visiting assistant, visiting researcher, or degree-based research that is from Indonesia, but at least how you could support also supervise but from Indonesia, but you could help us how to get the degree of the degree-based research of the researchers in Indonesia. I think the main purpose of this FGD today is about that. So before we develop together the research project proposal, it's important today we can have a clear map of our potential synergy among the two platforms on superfood, alternative food, or precision agriculture. And the second, how we can also have kind of big idea or concept for the collaborative research. And it is also hopefully that we can have joint publication, joint IP based on these two platforms. And it's the important thing that I would like to deliver in this speech. I hope we could continue our collaboration, and after this moment, after this FGD, this event, we will continue, we will follow up with the next meeting with each of you. Thank you so much.
Okay, thank you, Bu Punji. So we hope it's more clearly about the two platforms that will be open to doing the collaboration. And maybe I will next for the discussion. If you have any question or comment about the, especially about the two platforms that are presented already by Pak Yudis, Pak Dr. Yudhistira, and also Pak Satrio. Is there any, please? Please note me if I have any.
Good afternoon. Can I have a question to Pak Satrio?
Okay, sure, please.
Oh, thanks. Assalamualaikum warahmatullahi wabarakatuh. I think Pak Satrio presentation was very interesting because his presentation showed about the alternative and any possible superfood that can be developed or originally from Indonesia. And I like your explanation about tempe from Prof. Sastia. I know tempe is kind of like it's a big dilemma for Indonesia. We know tempe is like a food for people in Indonesia, but the biggest problem is the soybean is till now is still imported from another country. We cannot fulfill our soybean needs for either tempe or tofu production in Indonesia. But one that I'd like to comment about Prof. Satrio's presentation is about the how we develop superfood and different type of superfood in Indonesia. I would like to bring back all to before 70s, which is any Indonesian went to school around that time will be told that the people in Papua and Maluku, they eat sagu, and then people in Madura, they eat corn, jagung. But now none of Indonesian eat this as a staple food, and people in Papua and Maluku, they eat rice, and also in Madura eat rice. So by developing a new or introducing a new superfood is quite challenging because we need to do like, I call it anthropology study, how's their perception about any alternative food that they consume today, like staple food, all Indonesia, I will say we eat rice, but yeah, if you're gonna bring back to they eat corn or they eat sagu or any alternative, it's gonna be I'm not saying rejection from the people, but then it's quite challenging. Maybe we can convert the the look of the food, like make it look like rice or like instant rice made of either sagu or from cassava, or now very popular from porang. And have you considered any anthropology study or like social study for people perspective about alternative for rice? Because this group is mainly on developing a new staple food or superfood, but we need to know whether this product will be accepted and consumed by the people. I think that's my question. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi. Thank you, Pak Mulyoto. Pak Satrio, that is right.
Yeah. Okay. It's quite challenging to answer also because you talk about anthropology. But I think in the perspective of technologies, one of the idiom, if you don't eat rice, you don't eat. It's Indonesian idiom in some years related with the staple food. That's why in one of example of the food alternative, in this case carbohydrate resources, we propose about how to convert it into rice analog with various kind of carbohydrate resources. In this case, we try to use cassava. Maybe we can also use sagu or maize as the resources to convert it into the rice. But for the new generation, maybe not rice, maybe into the noodle also will be interested to make or to convert the staple food into that product. So I think it's interesting to see how to anthropologists research about what is the preference of staple food for Indonesian people. But you know, as you mentioned also, everyone now in Indonesia want to eat rice, not eat the previous staple food in their area. For example, we cannot find many people in Madura eat corn anyway. Maybe some older people, but for young people, not anymore. In Papua or in Maluku, we also cannot find younger people eat sagu anymore. But that's another problem. But I think yes, it will become our consideration in this project. But at least two kind of the product of carbohydrate resource or staple food in our consideration is rice analog or noodle. That's the common people like it. For the medium old like us, like me, maybe rice is preference, but the younger one, a noodle is preference. But the youngest one, maybe bread is preference. So maybe we will, as a technologist, we will convert the staple food resources into that product. But anthropologists research will be consideration. But for now, we will start from that. Thank you, Pak Mulyoto, for your
Thank you,
suggestion.
Pak Satrio. Yeah, but please remember that don't talk about noodles or wheat products because Indonesia don't have produce that. Indonesia don't have wheat or gandum. So please, look at like if you're gonna make analog, just make it from whatever available in Indonesia. Because like I know there's many possible replacement for staple food instead of rice. But we need to look at closely like some food that been abandoned like cassava and then porang. And I think we can find so many others in Indonesia. Thank you.
Actually, for noodle, we also develop the noodle product without wheat. We already produce the gluten-free noodle without wheat. Some produced from maize, some produced from cassava also. We already has a product and patent for noodle without wheat. So we can develop more from that perspective.
Thank you.
Okay, I think it's clear, yeah. So it's clear the diverse of the, yeah, we have many things that we can do in the superfood. Okay, is there a next? Is there any comment or feedback of the two? If I said not, may I ask Pak Philip, do you hear me? Dr. Philip here? Sorry, I have to spot your name. Yeah, here. Hi. So please, yeah, may I ask some kind of comment or feedback to that? We will launch here that yeah, Puji has already mentioned that we have will be launched the two projects. First is about the precision farming, and secondly about the superfood. So I think CCCAP is also related with this because the potato also is the one of the major important thing also in the communities in here, I think. So please, thank you.
Yeah, thank you. Can you hear me okay? Or?
Yeah. Yes.
You can hear me?
Yeah. It's clear.
Okay, good. Well, so first of all, I'd like to say thank you for inviting me to participate in this seminar. Actually, it's been very valuable to learn of the work that's taking place and the opportunities for working together with Indonesia. You know, currently we are developing a project together with BRIN, as you are aware. And that's been a great opportunity to actually understand a lot of the capabilities and capacity that BRIN has in this arena. So regarding these two areas, regarding superfoods and the opportunities for development of new precision agriculture, metabolomics, you know, I think from the perspective of the International Potato Center, so my base here in Beijing, or CCCAP, this is an area that we have a lot of interest in terms of precision agriculture for developing countries. And this is part of the reason that I'm participating in this call now. So I think that the opportunities exist to actually increase the efficiency that we have in agriculture. So I'm very pleased to see some of these the superfoods that you've been referring to, and you know, with the last person, the speaker that we had, we were talking about some anthropological studies and alternatives to rice, and of course, I'll come at this question with my own bias, maybe, in terms of the application of potato. So we've been talking about alternatives to rice, or using noodles, or alternatives to wheat, maybe. Something here that we frequently see is that there will be a rice that's actually coated in alternative crops as well. So here you can get rice that's coated in sweet potato or coated in potato. And of course, the objective there is to provide a different taste, but then also to increase the type of nutrition that's available. Another example that I can think of is when it comes to, I think sometimes following the trend of people can be valuable, but also my observation in terms of some of the work, some of the projects that we have in East Africa, especially working with orange flesh sweet potato, which is high in beta carotene, which is useful for nutrition in especially younger people. is that sometimes the local people actually don't like the food that would be nutritious for them. And so something that happened at one point in the history of the International Potato Center was that we introduced orange flesh sweet potato into East Africa, very nutritious, but people just didn't like the orange color. And so instead they would prefer white flesh sweet potato. And so what we did is that we started a marketing campaign. And as part of that campaign, we actually we did songs and dances and on TV and these types of things. And we also introduced how that it's possible to improve children's nutrition if mothers were to purchase the orange flesh sweet potato rather than the white one. And slowly, steadily over time, we actually changed the way that the families would purchase food and then produce it, prepare it in the home as well. So I think that there are different ways of approaching this. I think that it's possible to come at this with studies that actually what do people like, but then also I think it's possible to change it as well. For my own research here in Beijing, I think precision agriculture is something that absolutely I would like to follow. And that is actually leading a lot of my interest in working together with BRIN as well. When I had the opportunity to visit there several months ago, I was very impressed by the TEM and SEM microscope facilities that you have there. Actually, surprised is maybe the right word. And so actually that sort of motivates me more to want to be able to work together with BRIN in the future. Which leads me to the second platform idea, the opportunities for working together with BRIN. I think you talked about potential opportunities or in kind. I think that was the word that was used. So I think that I would be happy to provide support in whatever form that might look like. I think that's probably open to discussion and dialogue, and potentially, giving some advice or working together with colleagues in BRIN, and opportunities for maybe visiting it sometimes and something like that. So yeah, so I'm very happy with this opportunity, and thank everybody for the great presentations that they provided. Thank you.
Okay, thank you very much, Pak Bill. Yeah, I think we comment about the related with the good agriculture practices actually. So we have to make good resources actually doing with the good agriculture practices like the maintain of the disease or something kind of the stress, I think. So that one that maybe we can ask collaboration and another to create the collaborative of the approach, I think. So thank you very much, Pak Bill, for your comment, and yeah, we hope we can get some kind point to doing the collaboration. Okay, thank you very much.
Thank you so much. Thank you.
Okay, so is there any next from here? It's okay. Just raise hand. We have the two of postdoc actually in here from Research Center of Horticulture. May I have some comment or maybe like what Buji testimony during, yeah, we have the two postdoc here, and then maybe you need the testimony. Yeah, you can introduce yourself. Not mine.
Assalamualaikum warahmatullahi wabarakatuh. My name is Koko Dwi Sutanto. I'm from Central Java, Brebes. I'm here joined in BRIN since October 2024 until right now maybe it's about 8 months. Yes. In my background is about entomology and microbiology as well, about insect virus and about the IoT sensor. Maybe next.
Assalamualaikum warahmatullahi wabarakatuh. My name is Ishan Ali and I am from Pakistan. I am postdoctorate in Research Center for Horticultural Crop and my expertise in plant breeding and genetics. I am working under supervision of Dr. Tri Joko. Yeah. That's what? The Islam University of Bhavalpur. Yeah.
What do you think if you, during your join in this research center or ROAF? So I think, because we have the two of the postdoc, right, Puji? Okay, is there another question or comment? I think, is there any part of here? I think you want to say something? Okay, I think that's all the all discussion that we have. So thank you for the vibrant exchange, the good discussion, and I would like to highlight the points between how this FGD today, this session. So the first, the metabolomic as a tool to valorize the Indonesia food diversity. That is from Prof. Sastia, and then the AI and precision technology to revolutionize the farming, supporting the management for crop, water, soil, and harvesting management. Is it from Prof. Mahmud Iwan, and then scalable precision agriculture models that are also presented by Dr. Yudhistira. And the next, superfood and alternative to combat malnutrition. So the point, the next is the interdisciplinary collaboration is critical bridging metabolomic, AI, and on-ground agriculture. So policy, industry alignment to scale innovation like superfoods and also precision technology. The inclusive stakeholder engagement, especially, and also the local community. So the outcomes of 2 days of FGD will enrich the roadmap of the food synchron Nutrifoodsync program. So please let's continue this conversation beyond today to do the real collaboration. Finally, on behalf of ROAF, thank you. Thank you so much to all of the speakers, organizer, and all participants. So stay connection to follow up of this meeting. Thank you very much.
Thank you very much.
To do the real collaboration. Finally, on behalf of ROAF, thank you. Thank you so much.
Okay, we are so sorry about the technical issues. Thank you very much, Dr. Dunita Wikanutami, for skillfully guiding today's discussions. Ladies and gentlemen, as a token of appreciation, we will present the certificate of appreciation.
To the real collaboration.
That will be presented by the organizing committee on the screens. The first appreciation goes to Associate Professor Sastia Pramaputri, Osaka, from Osaka University, Japan. Wikanutami for skills. And the next certificate of appreciation goes to Associate Professor Dr. Mahmud Iwan Solihin. That will present UCSI University, Malaysia. And also, the appreciation is For Dr. Satria Krido Wahono, PhD, the head of Research Center for Technology and Food Processing BRIN. And the last but not least, we are going to give appreciation for our on-site speakers and also the moderator. And may we invite Mrs. Puji as the head of Research Organization for Agriculture and Food. Kindly present the certificate. Please come forward to give the certificate to the Dr. Yudhistira and our moderator for today, Dr. Dwinita. Thank you very much to Dr. Yudhistira Nugraha for the inspiring presentation. And the next presentation and the next appreciations goes to Dr. Dwinita as our moderator today. Please give a round of applause to our inspired and our speaker for this event. Please big round of applause. Well, ladies and gentlemen, on behalf of the organizing committee, we extend our deepest gratitude to all of our distinguished speakers and our moderators and also the participants that have been joining this event from the start and to the end. We also sincerely apologize for any inconveniences and also any technical issues during this event. It is our hope that the knowledge and discussions shared today will spark stronger collaborations and innovative solutions for sustainable food security. And also we kindly remind to all the online participants to fill the attendance form that has been shared by the organizing committee on the chat box. Well, ladies and gentlemen, as we close to this event, allow me to share a short rhyme as the token of our shared spirit and commitment. All in Indonesia, it is called as pantun. From superfoods to fields within, with mindful hands and caring heart, alternative crops, new hopes will send to guard our food right from the start. Thank you very much once again for your participation and see you next again on the next reference. Wassalamualaikum warahmatullahi wabarakatuh.